Well, hello, and I'd like to thank the organisers for inviting me back to Istanbul. This is about the fifth time I've been here, which is a measure of how fond I am of the city, the university, and above all, the people in Istanbul, who always looked after me very well, and I'm very happy to be back again. Do I have a laser pointer? Well, this is the campus in Edinburgh University, just south of the city where I'm based, and I want to give an overview of what 14-PC proteins are, what they do, and okay, good. And their role in neurodegenerative diseases, but given the nature of the conference, I'd also like to say something about their role in regulating gene transcription and gene regulation as well. So I won't be able to cover everything, but I'll give us a flavour of what 40-PC proteins do. And one thing they're involved in, which will be the focus of the talk, is protein aggregation. And so the main focus of my research is these proteins, including this 14-PC family of proteins, and they interact with proteins in the cell involved in Parkinson's disease and other neurodegenerative diseases, including CJD, Alzheimer's, and so-called a family of polyglutamine-repeat diseases such as Huntington's, and I'll explain where 40-PC is involved as we go along. Now, to begin with, in many neurodegenerative diseases, proteins accumulate in specific areas of the brain, abnormal forms of the protein which are misfolded, aggregated, forming fibrils, and that results in degradation of brain function because the proteins are not destroyed and turned over and degraded in the normal way, but they accumulate and affect brain function. And a common feature emerging is the involvement of this 14-PC family members with interaction proteins in many of these diseases. So, if we can show how these protein complexes form, how they function, and it may be able to devise means of preventing these proteins building up in the brain and hence producing therapy which might help prevent neurodegeneration. So this may lead to new therapies to use in research on how these protein complexes regulate brain function. So, protein aggregation is very important in neurodegenerative diseases, and most evidence points to the fact that the most toxic species of these proteins are not the highly aggregated plaques or tangles and that we can visibly see in the brain, nor the amyloid very high molecular weight forms which are relatively inert. It's the protofibrils or the oligomeric forms, the smaller forms, which can move around in the brain and pass on their ability to misfold other proteins. So these proteins may become covalently modified or proteolytically modified, forming small oligomers which can move around and infect other parts of the brain. Eventually they will form very large amyloid plaque deposits which are relatively inert. And what I will focus on is the role of 40-PC proteins in binding to some of these proteins and helping to cause bad effects in the brain. Now, there are many forms of so-called amyloidosis, or the ability of proteins to misfold into amyloid, and these are proteins involved in Parkinson's disease, including alpha-synuclein, but some other proteins I'll talk about, Alzheimer's disease, a beta peptide or tau protein, prion proteins in the CKD and other spongiform encephalopathies, prion proteins, And tau protein is frontotemporal dementia, amyotrophic lateral sclerosis, the superoxide dismutase protein which becomes misfolded, and so on. And there's a whole number of proteins where a CAG codon for glutamine becomes repeated, and this causes the protein to be unstable and to misfold and build up in the brain. And there's a whole range of these conditions which I'll talk about in more detail later, and some other dementia. But it's not only brain proteins which can misfold. In many other proteins, in many other conditions, outside. So, just to finish this one, the key feature of many of these diseases is the accumulation of these proteins in specific areas of the brain, which results in degradation of brain function. So, many other proteins in other parts of the body can also misfold. Other proteins, other polyglutamine proteins can expand and misfold, cause disease, and a whole range of other proteins, including proteins involved in type 2 diabetes. In fact, it's been shown that almost any protein can be induced to misfold and aggregate and accumulate in the brain or other parts of the body under the wrong conditions. So, what causes these proteins to misfold, and what helps them misfold, deposit, and not turn over, not be degraded normally? And as I say, 40pp proteins are involved in many of those, helping to cause these conditions in many cases. So, proteolytic processing, as well as is involved in this misfolding. It's a very important type of post-translational modification. So, as well as a lot of propeptides may be removed from secreted proteins such as insulin, activation of serine proteases and cyanogens, but also other post-translational modifications such as phosphorylation. And the addition of phosphate to proteins is a very important aspect of protein interactions. So most protein-protein interactions in neurodegenerative diseases are regulated by both phosphorylation and by proteolytic processing. And the build-up of these protein aggregates in these diseases are all regulated by phosphorylation and by proteolytic processing. And that's also including prime protein as well. So where does 40pp come in to this story? Now, as mentioned already, proteins associated with specific isoforms of 40p3. There are about 7 known distinct mammalian forms of this protein, and we'll look at the structure of this shortly. And they're involved in a range of neurodegenerative disorders. And the key feature, as I say, is the accumulation in specific areas of the brain in these proteins. And in many cases, these are involved with 40pp proteins. The proteins that accumulate are specific to each disease. They're not related to each other, but what the feature they have in common is that they 14p3 proteins associate with many of those. So what is 14p3 protein, and how does it associate with other proteins? Well, as you'll see shortly, 14p3 is a dimeric protein, and under the certain conditions, it can interact with other proteins. So it could interact, because it's a dimer, it could interact with 2 proteins on 2 sites, such as these blue and red proteins, and it can induce these proteins to come together and associate with each other, in this way behaving like a scaffold protein. And in this way, most of the protein, what induces the proteins to interact with 14p3 is phosphorylation, where under the appropriate presence of a protein kinase, which adds phosphate groups to these proteins, this creates a binding motif to 14p3, and the proteins then bind to 14p3 and associate in a complex. Okay, and related to this, is that 14C3 proteins have also been shown to be involved in trafficking between the nucleus. If it associates with proteins in the nucleus, it can form a complex, and the protein complex can go out of the nucleus into the cytosol. The proteins, 14C3 can also interact with proteins here in the cytosol and induce them to associate with the plasma membrane. And also, I have some examples of where 14C3 proteins can induce proteins to traffic between cytoplasm and mitochondria, and this is involved in regulation of apoptosis. Also, 14C3 itself can be involved in causing conformational changes in enzymes, and it can activate or inactivate proteins. So an inactive or an active form of protein can be modified by a kinase, which causes interaction, promotes interaction with 14C3, which then changes its activity or its conformation. And in some cases, which is important for the neurodegenerative disease story, interaction with 14C3 can cause the protein to be more resistant to proteolysis, more resistant to degradation. So I'll talk about examples of these as we go. One thing that's very important, of course, is to look at the structure of 14C3 and how these complexes form. And in my previous institute in North London, we had very good facilities for doing X-ray crystallography, much better than these facilities, where the caption says, investigating the first in its hand by Röntgen rays of X-rays. And here's a primitive X-ray generator which shines X-rays through those in its hand, and his colleague is looking straight into the X-ray beam. So what's going to happen to his eyesight in a very short time, I don't know. But we had much better facilities than these, and that enabled us to determine the structure of 14C3 protein. So I'll talk about some features of the 14C3 protein, which is very important for its function and interaction with other proteins. So, as I said, 14C3 is a dimeric protein, and there's a dimer interface here and here. And differences in the sequences of the amino acids here will allow 14C3, 7 isoforms of 14C3, to form a limited number of dimers. Not every isoform of 14C3 interacts with every other one. There's a limited number of heterodimeric forms produced. It's a very alpha-helical protein, what's called a helix in the protein. And two other important features I'll mention just now is that there's a cleft here, very highly conserved here and here in the other isoform. And this is the basis we discovered for the interaction site of the proteins that interact with 14C3. This is where they interact. But there's also some other features on the outside of the molecule that I'll talk about later. So that's the structure of 14C3. And how does it interact with other proteins? Well, it was discovered also by us and other groups that the 14C3 isoforms bind to a wide variety of cell regulatory proteins. Protein kinases, protein phosphatases, which take phosphate off the enzyme, transcription factors, various enzymes. And the interaction is regulated by phosphorylation at a typical amino acid sequence motif, where you have a basic amino acid, a phosphorylated serine, and another residue, and then a proline, which forms a kink or a bend in the protein and allows it to bend and bind in to the 14C3, as you'll see. And there are some more details of the motif, which I won't go into too much detail, but basic amino acids are such as arginine, aromatic or hydrophobic amino acids, phosphorylated serine, and then one of these amino acids is a proline. And clearly, if this serine residue is not phosphorylated, the protein will not interact with 14C3 or will interact with very, very low affinity. There are some other modes of binding, such as at the carboxy terminus of a protein, there's a phosphorylated residue here and the carboxycanone protein. And there are some tight binding non-phosphorylated motifs that can bind into the 14-50 cleft. But mainly it's phosphorylation, mostly phosphoserine, but phosphothreonine can also be present as well. So, further studies, further X-ray crystallography enabled us to find details of how this phosphorylated motif here, the phosphorylated peptide with the proline residue, and the phosphorylated serine. Here's the phosphate group, and that binds very tightly into this pocket here, but not phosphorylated, because the phosphate group here specifically binds to lysine residues, arginines, and a tyrosine residue here. So the phosphate is essential for the interaction, and this is very important for its function, as you'll see. Just another view of from the top of the phosphorylated serine binding very neatly into the structure. And if I have time, I'll talk about what we're doing with this towards the end. Now, I mentioned 14-PC was a dimer, so that we could potentially have two phosphorylated motifs binding to it. This could be two phosphate sequences from two different proteins, or it could be one protein with two phosphorylation sites, and this is very often the case. So you have one protein with multiple phosphorylation sites binds to one dimer of 14-P3. And this led Mike Jaffe and others to hypothesize that you could have a dimer of 14-P3, and you could have a protein with a highly tight binding phosphorylated motif that binds here, and then once it's binding, a second site can get phosphorylated and promote binding to a second site, and he calls this the sort of gatekeeper hypothesis. So you could readily remove the phosphate from this site because it's more weakly binding, not so strong, and then it's still binds, the protein still binds here, and then have more flexibility about the regulation of the interaction. And you can also, as you'll see, something that turns out to be very important is that the binding of the second site can create conformational change, structural changes in the protein, which allows access sites accessible, which would not be would not be accessible if the protein were free. So it can promote a conformational change in the protein. And to cut a long story short, I've got time to go into all of it, but it's now been shown that well over 300 proteins can interact with 14-P3 isoforms. Some cases with specific isoforms of 14-P3 from mammalian systems, from plants, fungi, as well as eukaryotes, etc. So I'll just talk about a few of them. The phosphorylated proteins that bind are involved in a wide range of intracellular signaling, trafficking, cell cycle control, metabolism, ion channels, physiology, etc. The protein that functions that interact with 14-P3 include protein kinases, which put phosphate groups on, like phosphatases, take them off, transcription factors, involving gene regulation, receptors, apoptosis regulating proteins, diverse enzymes, and most interesting for us, proteins involved in neurodegenerative disease as well. So I'll give you some examples of those as we go. I'll mention one example in the role of 14-P3 in regulating cell cycle in response to DNA damage. And it's been shown by other groups that 14-P3 can help prevent cells replicating if they are damaged. And how this works is shown as follows. In response to DNA damage by radiation or mutagens, this can stimulate the synthesis of p53 protein, transcription factor, which enhances the transcription of one specific 14cc isoform called 14cc sigma. The 14cc sigma binds to a cell cycle cyclin complex in the nucleus, and this complex then traffics out of the nucleus into the cytosol. And so the cell cycle proteins are now in the cytoplasm, and then so they're inactive, and this blocks the cell cycle at the G2 phase. So effectively, it doesn't directly inactivate the protein, but it does take it from the nucleus to the cytosol, thereby removing its function, and the cell cycle arrest takes place in response to DNA damage. Other 14cc isoforms are also involved in that DNA damage can also lead to stimulation of RAG3 or ATM, which will phosphorylate a so-called checkpoint protein kinase, which phosphorylates and inactivates a cell cycle protein phosphatase, and also binds to it and removes it into the cytosol. It has been shown in proof of this that knockout mice, where this particular isoform has been knocked out, these mice do not have this cell cycle checkpoint, and they're very sensitive to radiation damage or mutagens and readily form tumours, because the sigma 14cc is absent, and therefore they can't respond to the DNA damage by sequestering proteins into the cytoplasm. Now, I've mentioned the importance of phosphorylation on particular amino acid sequences to promote binding to 14C3, but we also showed that 14C3 isoforms themselves can be phosphorylated. And I won't go into too many details, but there are a few interesting phosphorylations on 14C3. So here's a generic sequence, a linear sequence of 14C3, and there are some phosphorylation on some isoforms which may regulate dimerization, because this is involved in the dimerization domain. There's some two isoforms can be phosphorylated at the carboxy terminus, including one isoform by the DCR or breakpoint phosphorylating protein, which is involved in leukemia and oncogenic protein leukemia. But I'll focus on a third isoform on the stephenine 185. I'll illustrate now. One example is the apoptosis stimulating protein BAD, or BAD, which can be phosphorylated on two or more amino acids, forming a binding site for a 14C3 dimer. Two phosphorylation sites, one 14C3 dimer. And this is in the cytosol of the cell, and it doesn't interact with the mitochondria and doesn't as the cell survives. But in response to cell stress or DNA damage, this during terminal protein kinase is stimulated. This can phosphorylate 14C3 on this residue I mentioned, and the phosphorylation then on the 14C3 causes dissociation from the BAD complex. So this can then bind to mitochondria and the BCL2 complex. This can then form a complex with the mitochondria and stimulate apoptosis or programmed cell death. So in response to cell damage, the net result is cell death, which is what you want, because you don't want a damaged cell to continue. Another aspect of response to cell stress is as follows. 14C3 binds to a number of other proteins, such as the so-called forkhead family of transcription factors. Now normally, this protein resides in the nucleus, and because it has a sequence on the surface called the nuclear localization signal, nuclear localization sequence. This means it's associated with the nucleus. But when it gets phosphorylated, it binds to 14C3, and the 14C3 physically masks the nuclear localization signal, and it means that the protein complex is found in the cytosol. Similarly, the c-Abl oncogene kinase also has nuclear localization signals and also binds to 14C3 and is localized in the cytosol. But again, in response to cell stress or daisy damage, soon internal kinase is activated. It then phosphorylates 14C3, dissociates from the complex, and now the nuclear localization signal is unmasked and free, and so the nuclear localization signal on this protein binds to important proteins, and so it's imported back into the nucleus. Also, similarly, the c-Abl protein dissociates from 14C3, and this also goes back into the nucleus. And the net result is that both the c-Abl activates genes involved in apoptosis, cell death, and the Foxhead protein also can stimulate. It's a transcription factor which will transcriptionally activate certain proteins and stimulate apoptosis. So, and that's the summary of it. I'll move back to neurodegenerative disease now, and where does 14C3 become involved? Well, in prion diseases, such as CJD, 14C3 proteins have been shown to be biochemical markers in the cerebrospinal fluid of proteins with these neurodegenerative diseases. This is first shown by a two-dimensional protein mapping, where two spots of protein here, you see them in more detail here. These proteins are absent from spinal fluid of healthy subjects, patients, but present in patients with CJD. And we showed that these two spots correspond to 4 isoforms of 14C3 in the spinal fluid of sporadic CJD patients, but not in healthy subjects. And this is a Western blot of showing the isoforms present in CJD patients, but not in healthy patients. So this is in detail. This has some functional relevance because these specific isoforms also appear in spinal fluid of sheep with scrapie and cows with BSE or bovine spongiform encephalopathy or mad cow disease as well. Now, 14C3 isoforms can also be found in spinal fluid of patients with other diseases, such as a particular isoform, a theta isoform in Alzheimer's disease patients' spinal fluid. So how might the proteins be involved functionally here? Well, in prion disease, we showed that in scrapie-infected mouse brain, as a model for sheep CJD, if you like, where there was severe pathology, we showed significant differences in 14C3 isoform distribution, and including co-localization in the brain of plaques with the misfolded form of disease form of prion protein with this specific 14C3 isoform. So the 14C3 isoform is binding to and helping deposit the disease forming misfolded prion protein. We showed localization of different isoforms of 14C3 in different regions of the brain depending on disease state, but I don't have time to go into detail on that. 14C3 is also involved in Alzheimer's and Parkinson's disease, and we showed that 14C3 co-localized with tau-rich tau protein neurofibril tangles. It's not a very good image of one, quite an old slide. Patients with Alzheimer's disease, and also in cases of Lewy body dementia, where also tau protein deposits occur. And we also showed that 14cc interacts with a protein called delta-catenin. On this phosphorylated motif that we said, this binds to a protein called presenilin-1, and this is encoded by the gene most commonly mutated in familial Alzheimer's. So the protein called the complex, which is presenilin protein, which predisposes patients to Alzheimer's disease. Now, we also showed that 14cc proteins are involved in a number of diseases, the so-called polyglutamine repeat diseases. And here, this is a case of proteins which contain polyglutamine stretch encoded by CAG, a glutamine codon, which is normally found in a few iterations of the CAG in normal patients, but in individuals with this polyglutamine repeat disease, this CAG codon is expanded from about 20 glutamines to about 40 in affected individuals. The best known of these is Huntington's disease, which I'm sure you've heard of, a late onset disorder where the CAG polyglutamine repeat is in the gene encoding a protein called Huntingtin. And Huntington's is inherited as an autosomal dominant disease. It gives rise to progressive and localized neural cell death. And this is characterized by voluntary movement, cognitive impairment, progressing to dementia, and mood disturbances. And the expansion of the CAG codon is directly involved in the disease because the age of onset of Huntington's disease is inversely correlated with the size of the expansion. So the longer the expansion of the CAG repeat, the earlier the onset, and the mean age is about 40 years, so it's rather devastating for the individuals involved. So, as mentioned, these expansions result in formation of neuronal intranuclear, in this case, the inclusions or polymers of the Huntingtin protein inside the nucleus, and they contain Huntingtin and the protein ubiquitin, which is normally involved in cell protein turnover. And they are most frequently observed in regions of the brain which show particular sensitivity to the Huntington's disease mutation, and most abundant in brains of juvenile onset Huntington's cases, where you have a large CAG codon repeat. And it's not clear just how this arises or why cell death is confined to particular neuronal subtypes, which it is, because the gene is widely expressed, but it's particularly localized in these particular type of neurons. But Huntington's disease is only one of 9 known neurodegenerative diseases caused by this type of mutation. So they include a disease called dentatoroubropallidoluysian atrophy, spinal bulbar, also muscular atrophy, which CFP proteins for CAG repeat in neuromuscular disorder. And there's a range of spinocerebellar ataxias, 1, 2, 3, 13, 17. We've been looking at the spinocerebellar ataxia 1, and that's what we know most about, the mechanism of how this occurs. And just to remind you that the proteins involved in these diseases are all quite distinct. The only feature they have in common is the ability to repeat the CAG codon by some pure mechanism. The mutant proteins are unrelated except for this polyglutamine tract of the CAG codon. And that's, so the aggregated polyglutamine seems to destabilize the protein and cause them to become insoluble and aggregated and build up in a proteinaceous protein deposit in the patient's brain for all these diseases, and they cause neurodegeneration. So the molecular mechanism undergoing this mutation involves the expansion and intergenerational instability of a tandem array CAG expansion in normal individuals, and the exact mechanisms of the neurotoxic mechanisms have been elusive. And normally, for these diseases, you have intranuclear inclusions, and the protein misfolds, interferes with DNA transcription and RNA processing. It can cause activation of apoptosis, dysfunctional cytoplasmic elements have all been involved in the process. The end result is apoptotic cell death, many aspects of neuronal function being disturbed in the rather devastating diseases. So, to focus on the cerebellar ataxias, so there are a heterogeneous group of severe late onset disorders caused by loss of balance and coordination, and most of these exhibit this autosomal dominant pattern of inheritance, and they form nuclear and cytoplasmic inclusion bodies, and the role of these is unknown, but the protein aggregation is a common step in pathogenesis of these otherwise rather heterogeneous disorders. The protein that's involved in spinocerebellar ataxia 1 is caused by the CAG repeat expansion in a protein called ataxin 1, and you're looking at that. So, where does 14C3 come in? Well, it happens that in a collaboration with the Zogby's group in Baylor, there's two isoforms, zeta and epsilon isoforms, can stabilize ataxin 1 when it becomes phosphorylated, and then it accumulates in the neurons, inducing this neurodegeneration. And this was published in Cell some few years ago, showing the co-localized 14C3 co-localizing with ataxin 1 and stabilizing it. So, just before we go back to spinocerebellar ataxia 1, we've also shown that another of these polyglutinin repeat diseases caused by atrophin 1, PLA, forms a complex with 14C3 isoforms, insulin receptor substrate, and atrophin 1 all interact, and the interaction is also in the phosphorylation site. And another group has shown that phosphorylated Huntington's, the causative disease of Huntington's disease, also associates with 14C3 when it's phosphorylated on an appropriate motif. Okay. So, what we know about the mechanism is that spinocerebellar ataxia 1, CAG repeat in ataxin 1, then ataxin 1 associates with 14C3 when ataxin 1 is phosphorylated on this motif of a basic amino acid followed by a phosphorylated serine, another amino acid, and a proline. So, without looking at the signaling response in detail, phosphatidylinositol kinase, which is associated at the plasma membrane, becomes activated. It then activates another protein kinase called AKT. AKT phosphorylates ataxin 1 on the appropriate residue. The phosphorylated ataxin 1 binds to 14C3, and this ataxin 1 is already fairly unstable, fairly insoluble, because it's already got these CAG repeats. And this seems to tip the balance between the ability of the cell to turn over and degrade and regenerate the ataxin 1, or allow it to build up. And so the balance is tipped with this extra association with 14C3, and the rate of proteolysis is slowed down even more, and the net result is that there's extra stabilization of the ataxin 1, and these polyglutamine repeat proteins accumulate in neurons, inducing neurodegeneration. And the 14C3 mediates this toxicity by binding and stabilizing the ataxin 1 and slowing its normal degradation even more. And so these proteins accumulate in a complex, and the net result is neurodegeneration. Right. How much time do I have? I think we're time right? 10. Okay, good. Right. Okay. We've also been looking at a number of other proteins involved in Parkinson's and other diseases. And proteins, I'll just go through this rather quickly because I've already given you an idea of how 14p3 might interact and stabilize these proteins. So there's a few other examples, such as in Lewy bodies, where the lesions in brain of patients with sporadic Parkinson's disease, they contain this protein called alpha-synuclein, and the alpha-synuclein builds up into fibrils. And alpha-synuclein has been shown to associate with 43 isoforms. And a lot of work from other groups, not from us, has shown that abnormally phosphorylated alpha-synuclein, as well as some of its mutants, are thought to be responsible for this apoptosis effect or programmed cell death of the protein in pathological conditions. And this occurs mainly in the dopaminergic neurons in the brain of Parkinson's disease, and for loss of dopamine and dopaminergic neurons and onset of Parkinson's. There's a protein kinase called leucine-rich repeat kinase, LRRK2, which is the gene for autosomal dominant form of Parkinson's disease. That interacts with 14p3 and is associated at particular areas of the plasma membrane called lipid rafts. And alpha-synuclein mutations also linked to this, because there's mutations in alpha-synuclein which disrupt the interaction of alpha-synuclein with these membrane rafts, and the LRRK2 can also cause this abnormal phosphorylation of alpha-synuclein. And another protein called Parkin ubiquitin ligase, this is a major cause of autosomal recessive juvenile Parkinsonism, and 14p3 beta isoform can bind to this ligase and suppress its activity. Normally, the ligase will ubiquitinate alpha-synuclein and allow the alpha-synuclein to be degraded and turned over, but alpha-synuclein can bind 14p3 from that complex and interfere with the regulation by 14p3 ether. And there's an image here taken from this endodermal paper, Sato et al. But 14p3 ether can bind to Parkin, especially in areas of cell stress, and block its activation, and so that the Parkin substrate, including alpha-synuclein, does not get ubiquitinated, does not get degraded by the proteasome. But by blocking the degradation and turnover of the alpha-synuclein, it can allow alpha-synuclein to build up its toxic fibrils and overwhelm the proteasome and cause neuronal cell death. So that's another way in which 14p3 can interfere with the normal turnover and help cause build-up of toxic fibrils in cell types. Right. Just to finish, it's obviously going to be very important to find ways to prevent 14p3 proteins from causing these protein build-up in the cell and allow the protein to be turned over and degraded normally. So one thing we've been looking at is ways of discovering how we can prevent 14p3 interaction with these some of these proteins involved in neurodegenerative diseases. And to that end, we have some very good facilities in our institute, wide range of state-of-the-art molecular biology, structural analysis, and we have the Centre for Translational and Chemical Biology, with protein production, characterization, screening for drug leads. We've also got state-of-the-art structural determination with NMR and mass spectrometry, a very good proteomics and mass spectrometry facility to identify proteins that interact with these proteins, including 14p3, and perhaps identify proteins in these disease aggregates. This is the website for the Centre for Translation and Chemical Biology, and one thing that was developed in Edinburgh by Paul Taylor and others is this Ludeus screening programme, Centre for Living Design at Edinburgh University, where we take the crystal structure, and we have the crystal structure of protein P3, and we know where these phosphorylated proteins bind, and so we know the contour surface, the acidic and basic residues, and the shape of the surface to which protein P3 interacting proteins bind. So we can make a contour map on the computer. Here's the peptide here binding. We can make a contour map like a contour map with a normal map, and you can see take information on the molecule that's known to be bound. In our case, it's a phosphorylated peptide from protein P3, and map on what we would predict to be interacting proteins or interacting molecules. So then we look to see where if other structures known in the database which might match this interacting pocket. Here's just a high-scoring ligand dock to another protein as an example. If it's a very good fit, it might match. And in the Edinburgh University database, it was 3.8, there's a lot more now, 3.8 million structures in the database, so we can match predicted structures of the potential binding compounds to the database of the known structures, so more than 4 million, I think. And then, once we have some high scoring potential ligands which might bind to this pocket in the computer structure, we can obtain samples of the compounds and test that they actually do interfere with the binding. And in the case of protein P3, here's protein P3 protein with an actual interacting protein binding to phosphorylated motifs. And this is one of the high scoring non-peptide binding components overlaid on the phosphoserine binding. So we've identified lead compounds that can fit into the site of interaction between protein P3 and its interacting proteins. And so we've screened using Ludeus in the in silico screening system. And then there's other programs we can use, such as protein P3, to look for prediction of protein P3 binding sites. And we have, as I say, the Edinburgh now has updated at least over 5.3 million compounds, or 3.8 million unique compounds. Some of them are repeats of each other. But this is the Edinburgh University ligand selection system, which can match in the computer structures of actual binding to putative silico binding. And we have a few lead compounds which might mimic the some of them are sort of nucleotide based, as you might with phosphate groups, as you might imagine. And then we'll be testing those to see if they actually do bind to protein P3 and actually do prevent other proteins interacting. And that's just the acknowledgements of some of the people who've been involved in the work and in the funding. Okay, I'll stop here. And, thank you very much for your
Thank you for your presentation. Now we will present your certificate of tooth sampling planted in your name in the Seven Trees Forest. Sayın Profesör Doktor Alice Stewart çok teşekkür ediyor ve adına diktiğiniz planların sertifikasını takdim ediyoruz.
Are there any questions? Nancy.
Thank you very much, Professor Etkin, for this detailed presentation. I'm wondering if some of, or is there any of some examples of these proteins which is used in routinely diagnosis at that moment as a biomarker of neurodegenerative diseases?
Yes, so the protein P3 in the spinal fluid of patients is used. Yes.
In which diseases?
In CJD.
Thank you.
Oh well, either my presentation has been very clear, or in which case there's no question, or I've been very, very obscure, and no one has understood. So either one or the other. Probably the latter. Probably going forward.
Şimdi 15 dakikalık kahve arası veriyoruz.
Makineye de vereyim ben.